Hello and welcome everyone to our last session in the GenScript webinar series. I'm Keshav Vasantavada, the Marketing Specialist for Protein Services here at GenScript. And I'm here to kick off our webinar today by introducing the presenter, Ray Wang. Ray is a senior scientist in the GenScript protein production group. She got her PhD in chemistry from Swiss Federal Institute of Technology, the ETH in Zurich in Switzerland. Her specialization was analytical chemistry. Ray has over 9 years of research and development experience in mass spectrometry and HPLC methods, especially in ionization source and HPLC method development. And in line with her background, Ray is going to discuss today the role of LC-MS in antibody drug discovery. But before we begin, a few housekeeping items. To ensure the sound quality of this webinar, all attendees have been muted. To ask questions, just type in your question in the chat box, and Ray will do her best to address them towards the end of the session. If we cannot get to the question during the session for any reason, we're going to make note of it and get back to you after the webinar. And I also want to let you know in advance that a quick poll will show up at the end of the webinar. It would be great if you could take a moment to provide feedback about today's session. Without any further delay, I invite Ray to start a presentation. Ray, it's all yours.
Okay, thank you. Thank you, Keshav, for your great introduction. Good morning, everyone. My name is Ray Wang. I'm a senior scientist in the protein production department at GenScript. Today, I will talk about the role of liquid chromatography and mass spectrometry in antibody and drug discovery. The topic includes 3 parts. First, I will introduce the LC-MS, including the basic information of liquid chromatography and mass spectrometry. Second part, I will introduce the application of LC-MS on the antibody and show some cases done on our platform. Finally, I will summarize the analytical methods of our platform. Now I will go to the first part. As we all know that the analytical part is very critical in antibody drug discovery. And this is the whole picture to show the analytical methods involved in the drug discovery process. And we can see that analytical methods are very important at each step of antibody drug discovery. Our LC-MS platform has very strong capability of chromatography, mass spectrometry, and electrophoresis. We can provide plenty of analytical methods for the antibody drug discovery. In this talk, I will focus on one analytical method, liquid chromatography and mass spectrometry, and show you what we can do with this technique. Liquid chromatography mass spectrometry, we call it LC-MS, is a sensitive analytical technique that combines the physical separation by high-performance liquid chromatograph HPLC and the mass analysis by mass spectrometer. Our platform owns a Shimadzu HPLC Nexura X2 and a Sykes TripleTop 4600. These 2 instruments coupled together can provide high sensitivity, high selectivity, good robustness, and LC-MS needs very, very low sample load. First, I will briefly introduce the specification of HPLC instrument. What we use for mass spectrometer is ultra-high performance liquid chromatography, so-called UHPLC. We can call it as the advanced version of traditional HPLC. The advantages of UHPLC over HPLC are low dispersion, lower extra column volume, high operation pressure, less sample load, better separation performance. This is quite suitable for mass spectrometer, which also requires very low sample load. And we use Nexura X2 from Shimadzu company. Next row, S2, is an all-around LC system like no other, and it is truly worthy of being called a next-generation UHPLC system. It uses revolutionary technology to offer high-performance, ultra-high-speed LC analysis and flexibility to allow configuring systems for a wide range of applications. It has maximizing analytical reliability, maximizing throughput, maximizing expandability, and high-performance units. Here I will briefly introduce how liquid chromatography can separate compounds. There are several types of LC separation. For the LC-MS of antibody, we often use reverse phase HPLC to understand the sequence or other structure information. On the top, the cartoon shows how the reverse phase HPLC works. The compounds pass through the HPLC column and they are absorbed on the stationary phase first, and then the compound will be dissolved to the mobile phase at certain time when organic modifier concentration reaches the critical value. Then it comes out the column together with mobile phase and detected by the UV. The most polar compound comes out first, and the retention time is least, as shown in yellow, and the least polar compound comes out the latest, as shown in blue. On the bottom, the picture shows the typical structure of stationary phase. It is very nonpolar with a long carbon chain. This chemical structure includes 18 carbon. Usually we use carbon 18 or carbon 8 column for the peptide and the carbon 4 column for proteins. Now I briefly introduce the theory of mass spectrometer. What is mass spectrometer? It is an instrument used to detect the molecular weight to charge ratio. The mechanism of mass spectrometry is to ionize the compound, separate the ions by mass to charge ratio, and finally detect the ions. This is the scheme of cordial time of flight mass spectrometer, which we use in our platform. The key parts of this instrument are the ionization source and the mass analyzer. There are several types of ionization source on the market. For example, ESI, MALDI, APCI, and APPI. There are also plenty types of mass analyzer, such as TOF, single quadruple, triple quadruple, quadruple TOF, Eintrap, and so on. Now first, I will introduce the ionization source. There are two types of soft ionization sources used for biomolecules. One is electron spray ionization, as shown on the left side, and the other is matrix-assisted laser desorption ionization. We call the MALDI. Our mass spectrometer owns ESI source. What does soft mean? It means that this ionization method can keep the biomolecule as its original status without any fragments, and we can detect the whole molecular weight of biomolecules with this method. The difference of these two sources are listed in the table. The operation environment of ESI is at ambient pressure, and the MALDI is operated in the vacuum. The ESI source is ionized compounds with multi-charges, while the MALDI source usually ionized compounds with single charge. The ESI source is very easy to couple with HPLC, but the MALDI source cannot. And ESI source cannot be durable of salt because salt can take away the charges from compounds, and the compounds cannot ionize well. Now I will introduce two factors of MS measurements for biomolecules. First, I will just briefly go through the theory of mass analyzer. What we use for our platform is time of flight, and here I will introduce the theory of the time of flight. This is an animation showing how the time of flight works. It separates different compounds through the flight. The time of flight is proportional to the mass over the charge, and that means the smaller compounds fly faster than the larger compounds as shown in this cartoon. Thus, the smaller compounds will arrive at the detector earlier than the larger compounds. And this mass analyzer can provide high mass resolution and mass ratios compared to the other types of mass analyzer. Moreover, it is suitable for all kinds of molecules, especially biomolecules. So in next slides, I will briefly introduce what is the mass resolution and the mass accuracy. This is the slide show what is the mass accuracy. The blue line represents the detected molecular weight, and the red line represents the theoretical molecular weight. The mass difference divides the detected mass is the mass accuracy. For example, if the theoretical mass of compound is 140 kilodalton, which is very typical molecular weight for antibody, if the mass accuracy is 10 ppm, this means that we can detect the mass difference as small as 1.4 dots. Our instrument has very high mass accuracy, which can reach 2 ppm, and this high mass accuracy allows us to understand the exact molecular weight and the post-translation modification of antibody. Next slide, I will show you what the mass resolution means. The mass resolution is the mass of the peak M divides the medium peak separation delta M. There are several ways to define the medium peak separation delta M in mass spectrometry. The two most widely used are the peak width definition and the valley definition. The valley definition defines delta M as the closest spacing between two peaks of equal intensity, with the valley between them less than specific fraction of the peak height. Typical values are 10% or 50%. In the peak width definition, the value of delta M is the width of the peak measured specified fraction of the peak height, typically 50%, which is called the full width at half maximum. And in our lab, we use Triptych 4600 mass spectrometer from Science, and this system delivers high resolution, accurate mass and MS mass for qualitative and quantitative workflows. I list the specification of this instrument. The mass accuracy is 1 ppm and the mass resolution is 25,000. The ionization source is ESI Turbo V ionization source, which has very good ionization efficiency, and it provides very good sensitivity and good robustness. Next, I will go to the second part. It's about application and case studies of LC-MS. The discussed application revealed LC-MS to be a versatile technique with the ability to determine many molecular and structural attributes of antibodies. These include analysis of molecular weight, post-translation modifications, conformation changes, binding interactions, and epitopes. Here we highlight several applications based on our LC-MS platform that have been already used in previous cases. These applications include molecular weight of heavy and light chains, sequence conformation, post-translation modification, and Glycan analysis. First, I'm going to introduce the molecular weight test by LC-MS. Here is the protocol how to do the measurement. We usually do 4 types of molecular weight measurements. there are intact molecular weight and glycosylation molecular weight, reduction molecular weight, and reduction and anti-glycosylation molecular weight. The sample preparation include the buffer exchange, the chemical reactions including the anti-glycosylation, reduction, and alkylation. The buffer exchange step is open at the end of this preparation, and the sample preparation procedure can be optimized depending on the sample. Here I will present a case study of an antibody including these types of molecular weight measurements. On the left side is the original mass spectrum we obtained by the instrument. We can observe plenty of mass peaks in the range of mass m/z from 2000 to 3500. This Gaussian shape peak distribution is caused by antibody ions with various charge numbers. The MS software can do the data analysis and deconvolute the mass spectrum to the spectrum of the intact molecular weight. We can see the peak around 149 kilodalton on the right side of the spectrum, and the smaller peaks around this main peak are those isotope peaks. Thus, we can know that the intact molecular weight of this antibody is around 149 kilodalton, which is very close to its theoretical molecular weight. Then we want to further investigate the glycosylation of this antibody. So what we do is to digest the antibody with PNG, which is a specific enzyme for glycan. I will introduce this enzyme in the later slides in detail. After we get rid of the glycan on this antibody, we do the MS measurements again and obtain the molecular weight without glycan, as shown on the right side of the top. Then we compare the intact molecular weight and the molecular weight without glycan. There is a mass difference around 2,900 dalton, and we can confirm that there are two positions of glycosylation, and the glycan is GLF according to its molecular weight. We can also use the reduction and alkylation to understand the molecular weights of heavy and light chain for this antibody. Here are the results. At the top of the slide is TIC spectrum of the antibody after reduction. We can observe two peaks around 10 to 11 minutes. These two peaks represent the light chain and heavy chain separately. The mass spectrum of light chain is on the left side of the top. The convolute spectrum is on the right side. The molecular weight of light chain is around 24 kilodalton by the measurement, and the mass spectrum of heavy chain is on the left side of the bottom. The deconvoluted spectrum is shown here. The molecular weight of heavy chain is around 51 kilodalton. These results can match with the data of intact molecular weight measurements. Molecular weight determination is also very important for biosimilar study. Here is one case of biosimilar study with molecular weight measurements. On the left side, there are the mass spectra of heavy chain, and on the right side, there are the mass spectra of light chain. We can see that the mass spectra of biosimilar are quite close to those of reference drug. And we can see that high-resolution mass spectrometer can allow us to do the quality analysis of biosimilar very efficiently. Now I will go to the second application of LC-MS on antibody. It is a sequence confirmation, which is quite critical for the antibody drug discovery. The sequence of the antibody product can be determined via a number of approaches. Our experts in GenScript perform sequencing studies using a broad range of enzymatic or chemical digestion with LC-MS mass spectrometry analysis. we offer to make a carefully designed sequence confirmation of the antibody. Based on the sequence of a specific antibody, we will select 2 to 5 digest enzymes for the experiment. It will ensure that we can get almost 100% sequence coverage. The procedure of the sequence confirmation experiment is shown in the scheme. We first digest a sample with 2 to 5 enzymes. Then we perform peptide mapping of digested antibody. We use MS analysis so we can understand the detailed information of each peptide. Finally, we obtain the results based on the database and compare to expected sequence. This includes comparison with a number of modifications. Here is one example with antibody. We separate the trypsin digested peptides by reverse phase HPLC, and peptides were identified and analyzed by MS. This is the TIC spectra of MS and MS analysis. By analyzing the MS data, we achieved 100% sequence coverage of client sample. Sequence confirmation is also very important in biosimilar study. It is the first step of biosimilar development because the sequence of biosimilar should be exactly the same as that of the reference drug. We have done plenty of biosimilar cases with 100% sequence coverage. Here is one example. This is a mass spectra of peptides after digestion. We can see that the spectrum of biosimilar is very close to that of the reference drug. According to the data analysis, we also confirm that the sequence of the biosimilar is exactly the same as the reference drug. We can also do the measurement of N-terminal sequence. Here is one example. The client wants to know which N-terminal sequence belongs to their sample. So we do the same suggestion procedure and analyze MS data in the database. And we found that the fourth peptide sequence has the highest match, and it is probably the N-terminal sequence of the client sample. Then we analyze the MS data according to the sequence to check whether the molecular weight of fragments by the sequence can match on the peaks of MS spectrum. The results show that this N-terminal sequence is the one of the client sample. Now I will introduce the third application of LC-MS. It is the glycan analysis. There are many different kinds of glycosylation for protein, but for antibody, the glycosylation types are much less, usually N-glycosylation and occasionally O-glycosylation. The typical N-glycosylation of antibody is shown in the figure. The glycosylation typically happens on the two heavy chains at 297 sites, asparagine in the Fc region. The glycosylation influences the function and the structure of antibody, including the complement activation, receptor affinity, and maintaining Fc structure stability and efficiency of the therapeutic MABs. This is the typical N-glycol form, and we use an enzyme PNJF to get rid of the glycan from asparagine residue. This is the structure of the PNJF. The cleavage site of this enzyme is highlighted by the red circle, and the reaction is shown at the bottom. When the glycan gets rid of the asparagine becomes aspartic acid. For glycan analysis, we use a whole protocol provided by Waters. Here's the scheme. This protocol significantly simplifies the sample preparation, allowing deglycosylation, labeling, and the sample cleanup to be complete within 30 minutes. rapid flow MS from waters is used as the denaturation reagent. And here is the case of glycan analysis. And we use UHPLC helix MS to identify the glycoform in antibody. The total ion current chromatogram shows the peaks identified for various glycans. The corresponding glycan for each identified peak was analyzed and listed in a table. In this case, we identified more than 20 glycans for this antibody. Glycan analysis is also an important part for biosimilar study. We need to compare the glycan profile of biosimilar with that of a reference drug. And here's one example of biosimilar we did in our lab. These are the overlaid HPLC spectrum of drug and biosimilar. The peak area percentage of each glycan is listed in the table. The results show that the glycans of drug and biosimilar are very similar, especially for those glycans with high contents. We also can do the N-glycan site confirmation by LC-MS. Here is a scheme of the whole process. The difference from the glycan analysis is that we will analyze the peptides with and without glycans after digestion by MS and MS measurements. Then we will do the database search and mass spectral comparison manually to confirm the precision of glycosylation. Here's one sample we did before. On the left side, it is the TIC spectrum of trypsin digestion. And on the right side is the case with trypsin and the PNGF digestion. These are the mass spectra of these two methods. The mass difference is around 1,200 Dal, and we can confirm that the glycan is MAN5 and the glycosylation happens on this peptide according to the molecular weight and MS mass data of this peptide. We also search other possible glycosylations on this antibody. So we checked the MS and MS spectrum of peptides with potential glycosylation. For example, these are the mass spectra of this peptide with potential glycosylation. We cannot observe any mass difference with these two methods, and this suggests that there is no glycosylation on this peptide. Now I will introduce the fourth application of LC-MS on antibody. It is the mapping of disulfide bond. Disulfide bond plays an important role in antibody drug. It has an important effect on the integrity of final product. Improper disulfide bond arrangements during the cell culture or purification process contributes to aggregation. And biosimilar guidelines from the US FDA and within the ICH framework, disulfide bond mapping is also seen as critical. Thus, investigation of disulfide bonds is very important in antibody drug discovery. This is the figure of a typical IgG antibody and the location of disulfide bond. The representation of the disulfide bonds was hold the heavy and light chains together are highlighted by a dotted circle. On the right side, this is the protocol how to identify the disulfide bonds by MS. It usually involves the reduction, alkylation, and digestion. Here we introduce the protocol in detail. First, we determine the numbers of disulfide bonds in antibody by reduction with DTT and alkylation with IAM. According to the mass difference caused by the reactions, we can know the numbers of disulfide bonds. Then we digest the antibody and do the reduction and the alkylation to identify the location of disulfide bonds in the sequence. We'll do the database search of MS data and check the results manually to confirm the location of disulfide bond. Here is one case to identify the disulfide bond in the antibody. On the left side is TIC spectra of the sample before and after the reduction. There is one peak disappear after reduction, which suggests that there's disulfide bond on this peptide. On the right side, these are the mass spectra of the peptide before and after reduction. We can see that all the MS peaks disappear after reduction, which confirm that there is disulfide bond on this peptide. Then we further analyze the MS and the MS mass spectra of this peptide, search the database, and confirm the structure by checking each MS peak manually. We found an intern disulfide bond in these peptides. Now I'll go to the final part. I will summarize the whole talk. Our LC-PLET MS platform can do all these tests with mass spectrometry, as I mentioned in the previous slides. And these tests are very important in antibody drug discovery. We are also developing new methods and techniques for the antibody, and we will release test surveys of LC-MS next year. Moreover, our platform is not only restricted in the mass spectrometry aspect. We are also proficient in LC methods and capillary electrophoresis methods, such as CIEF and CESDS. Here's the whole picture of analytical methods we provide on our platform. And the GenScript can perform a variety of characterization testing to ensure integrity of the final product. And finally, thank you for your attention. Now we can go to the question part.
Hi Ray, thank you for that wonderful presentation. Are you able to see the questions panel?
Yeah, and I'll just check the question now.
Okay, go ahead.
Oh, there's one question, and that is, what is the advantage of using an Orbitrack over top analyzer? On our platform, we own a triple top mass analyzer, but for the Orbitrack, I think they also have very high mass resolution, which is close to the triple top. So they have very similar behavior according to the mass resolution and the mass accuracy. So they can also do for the antibody study.
Right. Thanks, Ray. So I have a few questions. Sort of, you could think of them as frequently asked questions in the LCMS area. So can you tell us what kind of software you use for the MS data analysis?
Okay. On our platform, we use the protein palette and the biofarm view to analyze the data, which belongs to the science company.
Okay. Okay. And so what is the sample load for the molecular weight measurement, and is there any requirement for the sample?
The typical sample load for the measurement should be higher than 200 microgram at 0.5 milligram per milliliter. And also there are some other requirements for the sample. For example, there is no detergent in the buffer system of the sample, and you also need to provide information of the sample, including the buffer concentration, the theoretical molecular weight, or the sequence of the sample.
Okay, okay. And what about samples that are easy to aggregate? Is there any special requirement for such samples?
It's better to clarify any sample information, because if the sample is easy to aggregate, we have to deal with low concentration to avoid aggregation during the measurements.
Okay, okay. And what is the sample load for sequence confirmation, and is there any particular requirement for that kind of sample?
Yeah, the sample load for the sequence confirmation should be higher than 1 milligram at the 0.5 milligram per milliliter concentration. And also you need to provide the theoretical sequence of this sample and the concentration and the buffer. And there's no detergent in buffer system for this sample.
Okay, okay. And so what about sample load for N-glycosylation site determination?
It is the same as the sequence confirmation is higher than 1 milligram. And also you need to provide the basic information of this sample, including the buffer concentration, molecular weight, and the theoretical sequence.
Okay, okay, great. So, about the instrumentation, can you specify what the mass to charge ratio of the instrument is?
You mean the mass to charge ratio range, right, of this instrument?
Right.
For this instrument, it's from 50 Dal to 4,000 Dal.
So 50 to 4,000 Daltons, you said?
Yeah, yeah, exactly.
Okay. And so what enzyme do you typically use for the digestion of sequence confirmation?
Usually we use like 2 to 5 enzymes, typically like trypsin, chymotrypsin, glucine, asparagine, and like that.
Okay, okay. And this was a question that I always had back in graduate school as well. So how many amino acids typically can you identify using N-terminal sequence confirmation?
Typically around 20 amino acids or less, but the exact numbers depend on the sample sequence.
Okay, okay. And so what other types of post-translational modifications can you detect using mass spectrometry?
Actually, we can also do like the ionization, methionine, oxidation, C-terminal lysine variants, but actually these tests are customized service, so it's not like a regular service. as disulfide bound analysis or Glycan analysis.
Okay, okay. And so what about, I know you think you addressed the N-link glycosylation. What about O-link glycosylation site determination? Can you do that as well?
At our current techniques, we cannot do the glycosylation.
Okay, so is that like a technical like issue there with the determination?
Because it's the instrument issue, because we have different um, techniques for the MS analysis, which is not suitable for all glycosylation.
I see, I see. Okay, okay. Great. Thanks, Ray. So are there any other questions on the question panel? Are you able to view it?
Here's one question mentioned that how to apply the LCMS to assay the microbiomass and how the sample preparation to assay. Actually, for LCMS, you can do the mass analysis for any kind of molecules. But I guess for the microbiomass, maybe we should use the different sample preparation, also different separation methods for this type of the molecules. So it's I think we can do with LCMS, but we never done on our platform. So based on the theoretical way, it could be done with LCMS. There is another question about the general enzyme protein ratio when we use the enzyme digestion in LCMS. In our lab, we usually use 20:1 ratio for the sample to enzyme. Here another question asks why we use 2:5 enzymes for digestion, because this enzyme has different cleavage sites for the protein. So in order to achieve 100% sequence coverage, so we usually use 2:5 different kinds of enzyme to ensure to cut all these sequence to the proper size peptides, so we can identify by the MS and analyze the data.
Great. Ray, thank you very much for that wonderful presentation and for answering all the questions. I don't see any other questions in the question box panel, so I think we can conclude the webinar here. I hope our attendees derived value from this webinar. I surely did. I don't have a very strong background in LCMS, so your review of the basics and helped me gain some more understanding of this powerful and versatile technique. So as we conclude our webinar, a quick reminder once again to our attendees, before you leave, and as I said at the beginning, it would be great if you could take a moment to complete a quick poll right after this session ends, to tell us how you like this presentation. Your candid feedback will also help us improve the quality of our services. So once again, thank you all for your time and kind attention. And on behalf of the entire team here at GenScript, we wish you all success in your research. Thank you very much.